Hello. Right, has that worked? Oh, is this a thing of beauty? Right, if we could not have any spam, so I can't bother to have to deal with any like moderation stuff. Right, if anyone can let me know that is actually working, that'd be handy. Beautiful stuff. Right, how are we feeling? Lovely stuff. Right. Okay. Right. I think I can even do a quiz. Oh, this is beautiful. Look at this. This is high tech stuff. Right. What topic? Right. We've got cells. Got a bit of cells. We've got organisation. We've got infection and response. And we've got bioenergetics, that's the one. Right. The poll, look at this. He's on form. Look, I've even got questions. I originally printed them smaller, like they're for ants. Look at this, huge. That's the one I want to tilt. Who knows? We'll find out, won't we? Just cells on top. This is nail biting stuff. This is nail biting stuff. What a way to spend a Monday evening. Right. I mean, next vote's going to win it, isn't it? Because I'm not dealing with a tie. Well, let's start with cells, shall we? Because why not? Bosh. Poll ended. Right. Decision made. Engagement. Right. Okay, so we were talking in class earlier about like the real basics when it comes to cells. Okay, and that's knowing the parts. All right. So, just for the sake of completeness, okay, some of you might be really happy with it. Others you might not be. We stereotypically draw a plant cell like a square because why not? And animal cells we typically draw as a circle. Okay, and they end up looking a bit like a fried egg. Lovely stuff. Now, plant cells have a cell wall. That's the big giveaway. We've got our permanent vacuole. Look at that, lovely. Nucleus, I'll do that. Yeah, I think she is. Oh no, not year 10. Not year 10. I think we're getting. No, she is. Hang on, let me look at my schedule. Yeah, lesson 1. Right. Look at this art. High art. I know what you're thinking. So, did you do GCSE art? I think the answer's obvious, isn't it? No. All right, we're going to go ribosome. Artistic. Right. So we need to be familiar with what the parts are and what they do. The major one, which I really want to draw attention to, is that one. That's the cell membrane. So many people get the cell membrane being on the outside of a plant cell. It's not. Oh, I just written plant. Long day. Cell wall. Okay. We only have a cell wall round a plant, or or yeast cell or bacteria. It's definitely not an animal cell. That's the main thing. They're made of things. For a plant cell, they are made of cellulose. It's because they're wheeler dealers are plant cells. They'll always sell you those. I'll run with it. Right, we've got our chloroplasts, and our chloroplasts are only in our plant cells. One of our 3 parts which are only in a plant cell. And crucially, these do photosynthesis. More on that in the bioenergetics unit. Okay, and we've also got mitochondria. Okay, they are also in our animal cells, and they do one thing, and that is respiration. Only in every cell needs to be doing respiration, otherwise it is dead. There's 2 types, aerobic, anaerobic. We'll come to that in a bit. We've got our nucleus, which controls the activities of the cell. Don't call it the brain of the cell. We've got our cytoplasm, which fills the cell. Most of the chemical reactions happen there. And our kind of other lurking one on the plant cell is our vacuole. Animal cells and plant cells both have their DNA in a nucleus. That makes them collectively something called eukaryotes. Okay, remember, you carry this in the nucleus. That's probably the best way of doing it. If my voice or the voice level or anything is off, just do let me know. We've then got the opposite of that, which are prokaryotes. Now, prokaryotes are much smaller, and they don't have a nucleus. Their DNA just chillaxes. Okay. Now, what's the problem with that is it means it doesn't keep it safe. It kind of like mutates quite a lot. It's much simpler though. And prokaryotes crucially have no nucleus, and they also have no mitochondria. And they have no vacuole, and they have no chloroplast. They have no what we call membrane-bound organelles. Right. Now, I would say arguably the hardest bit of cells is not this. This is our like real core, right? I just wanted to start on that. Okay. I just wanted to really start with that. But what I did want to kind of go talk about, and that's the transport. So that is the 3 ways that we can transport things into and out of cells. Anyone got any good snacks, by the way? I've got an epic bottle of water. I don't know, one of those ones where you think that's too big. That's what I've gone for. Being healthy, innit? Right. Okay, so there are 3 types of transport. We've got diffusion, we've got osmosis, and we've got active transport. Okay, now diffusion is where we go from a high concentration to a low concentration. So let's give an example. Let's say you are the alveoli in the lungs. Okay, and here's our little blood vessel. Lovely stuff. Now, in our alveoli, we have got air. This is blood. Now, in the actual air, we've got lots of oxygen. So we've got, oh, that's a fat end. We don't need that. I want a bit of precision. I'm a precise man. We've got high oxygen. And we've got low carbon dioxide. And in the blood, we've got low oxygen. It's just been used up. And we've got high carbon dioxide. So, hopefully, this might make you think, okay, diffusion, high concentration to low concentration. We go from the high O2 to the low O2. high CO2 to the low CO2. And we have now done gas exchange, where we've taken our air and we've kind of swapped it out. Sometimes I like to ask, how does this get better? And it gets better in a few ways. First of all, they're very thin walls, very short distance. That means that it doesn't have to go far. It's got good blood supply. So it means that anything which is kind of done is whipped away. And this is kind of the main big dog of this is the idea of a large surface area. Right? If I have, it's got to go through a surface. So if I have a, right, it's like trying to get out of a building. It was on fire. That's a poor example. But if I only have one door to get out of, it's going to be really slow. If I have many doors, I can like kind of leave really quickly. So it's the same thing here. If I've got lots of space, I can get in and out like kind of across the whole lot. Okay, so that's diffusion. Then we've got osmosis. Now, osmosis is basically the same thing as diffusion, but it's just for water. And it's a little bit confusing sometimes. So this is high water conc to low water conc. Now, this is why it is slightly confusing, because if I looked at a hi Jasmine, you don't need to worry about this yet. You're in year 8. Go to bed. You have in your cell, you've got lots of water, you've got lots of stuff in the water. Okay, so if I take my cell, right, inside here I've got, you know, some bits dissolved in water, and let's say I put the cell in pure water. So out here, there's lots of water. Inside the cell, there's water, but crucially, there might be some bits in there. Right. In this situation, right, I've got a higher concentration of water outside than I have inside. So the water will move into the cell. Okay. Now, the reason this is confusing, because at the moment I think hopefully you go, yeah, that makes sense. The reason this is a little bit confusing is because I don't really tend to fuss about the water, right? If I was making a glass of squash, the concentration in that refers to the squash, not the water. So it looks like there's stuff in the cell, and the pure water looks like it's a low concentration of other stuff, but it's a high concentration of water. All right. So when I'm putting it in pure water, the high concentration of water there is outside. If I was doing this with, let's do the same thing. And we'll put it in, but this time we'll put it in really sugary water. So I've got the same amounts inside. Now here in this case, I've got more water concentration inside the cell than outside the cell. So the water moves out. That means the cell would get all kind of, now that's technically called flaccid. And this one will get kind of pumped up and that'll become turgid. So this one becomes turgid. And this one becomes the flaccid. Okay, and then you can imagine a halfway point where it neither goes in or out or shakes it all about or does the okie dokie. In that situation, it's what we call isotype and it will remain the same. Okay, right, last one of the transport is active transport. Now, this is probably in a weird way the easiest one to understand because it's low concentration to high concentration. So it's going against the kind of instinct, right? This just happens just all the time. This is kind of you're forcing it the other way. Now, I like to think about this like the game Hungry Hungry Hippos, if you've ever played Hungry Hungry Hippos. Yeah. Am I really quiet, by the way? Or am I like the right volume? Because the live stream kind of bit makes it seem like I'm only like edging onto 2 green dots. I'm loud. Oh, I'll take that then. It's cool. Right, it's kind of like Hungry Hippos. Now, with Hungry Hippos, right? You've got like beautiful. I've got like the arena, right, with the little hippo. This is the worst drawing I've ever done. There's like the little plunger. Love that. And then inside here, you got all the balls. Right? Now, in your little container, you've got lots of the little balls that you've all collected. Okay? And that means here I've got high concentration, right? But I've got a low concentration of these, but I still want them. So, you know, you put in energy, you start like smacking the Hungry Hippo, he goes and he collects them all in, right? So you are going from low to high. But what did you have to do? You had to put in energy, right? You had to put in energy to do that. Where did that energy come from? It comes from respiration. So if you are a cell which wants to do active transport, you will have mitochondria in there. That mitochondria will provide you energy, and that means you do it. Now, what kind of things do active transport? The big one they always like to talk about is roots, right? Plant roots. So plant roots will do all three. This is why they like to talk about them. So they'll do things which have got a high concentration in the soil, will do diffusion. Water will do osmosis. And then if you've got like ions in the soil which are quite rare, but you want them into the plant, you'll do active transport. Okay, so your root hair cells, which look like Pinocchio, they have lots of mitochondria in them to provide energy to start absorbing that. And again, that big Pinocchio style nose, that's for our large surface area. Okay. Next thing to do with that might be your gut if you're absorbing stuff for from like your food and digestion. Should we look at some questions? Does anyone have any questions? Anything I've said so far? I'll give it like a couple of seconds while I just like have a sip. Fine. I'll take it, everyone's fine. Right. Here is. They have to be dilute concentrated. Yeah. So this is what I'm saying. This is why it's a little bit confusion, right? So a little bit. I am confusion. This is why it's a little bit confusing, is because it's high water concentration to low water concentration. So if I was looking at this in terms of like a glass of squash, right, this would look like a dilute glass of squash, right? Because that's a high water concentration, low thing. Yes, it is all in combined. Delare's just still hurt because there's no space physics, isn't she? And then here, right, that's a low concentration of like water, but a high concentration of your squash. So this, even though it's a low water concentration, you would call concentrated. And this, even though it's a high water concentration, you would call that dilute, just to answer that question. That's all right. You'll be armed with the beautiful knowledge. Yes, absolutely. Yeah, because when you're looking at it, you're only really fussed with like the stuff which is in it, right? Which is why it's sometimes a bit confusing. Right, let's try. Look at this setup. Whoa! Right, parkour. Shall I try and adjust this? Is that a bit better? That's a bit better. It's just bouncing all over the place. It's such a garage setup. Chill. Right, this is the classic osmosis practical, right? To the point, right? I just want to say they could use any type of cell, right? Just because everyone does it with potato doesn't mean you don't do it with potato. Can't do it with something else. One year they did it with carrots, and then they started lobbing carrots at the AQA headquarters, which I think is top top joke, but it shows that people aren't understanding it because it doesn't matter about what it is. But here we've got a student investigated the effect of different concentrations of salt solution on the mass of uncooked pieces of potato. Sometimes they ask actually why they use it uncooked, because if you cook it, all the cell membrane will burst. Osmosis needs to happen over a membrane. So if you haven't got a membrane, that won't work. Give 2 control variables the students use. So this is our method. I'll come back to it. Control means things that you are keeping the same. So if I look through this, okay, says cut 4 pieces of potato to the same size. So it's the same size of potato. It's one of them. Okay. Now, crucially, what it can't be, and this is a good point just to, again, I'm starting to try and chuck things in if I can, dairy milk ice cream. So the other variables are your dependent and your independent. And the dependent is what you measure, and the independent is what you change. So here, if the dependent is what you measure, that's what you're going to look at the other side, and the independent is what you have decided to change. Well, that's always given in like a preamble sentence, right? So it says the effect of different concentrations of salt solution. So that is going to be our independent, because they are changing the concentration of salt solution, and what effect that has on the mass of uncooked potato. So that is what we're measuring. So that's our dependent. So as long as you don't mention those, you're probably going to hit a control variable. All right. So we've got 2 control variables. We've said one is the same size as potato. You put one of the pieces of potato containing 100 centimeters. Okay. Now, centimeters cubed. Now, the concentration I can't touch, but I can say the volume of solution. Okay. Try not to use the word amount, or we do not like it. Okay. I think I'm slowly losing my mind. Right. The student needed to be sure the measurements were as accurate as possible. What should be done to each piece of potato after removing it from the solution and before measuring the mass? So this is really just another specific one about that practical, just to check that you actually know and remember the practical. If you remember, you just put it in solution. So if you get out and you're going to start weighing it, it's going to be unfair because it's got water all over it. So the first thing you're going to do is you're going to dry it. Name the piece of apparatus the students use to measure the mass of potato. Again, some people sometimes just overthink it. You're measuring the mass of something. Use a balance, mass balance, such a scale. Okay, right. Okay, so I've got certain results. They've done it. Here are the concentrations of salt solution. They've measured them at the start, measured them at the end. They've looked at the change, and then they've looked at the percentage change. So by doing a percentage change, you've accounted for what's going on in terms of like the starting masses and stuff like that. Now, we've got, what was the resolution? Resolution is kind of your intervals, right? So if I've got a ruler nearby, I think I do. Hang on. Right, here's a ruler. Now, if you have a look, the lines on this are 1 millimeter apart. Sorry if I keep wobbling it. Don't want to make you motion sick. But the lines here are 1 millimeter apart. So the resolution of this ruler is 1 millimeter. Okay. Now, if I look at these, right, can you see that all of these are measured to 1 decimal place? So that gives it away that the little lines were like 1 decimal place away. So that means the resolution is 0.1 grams. Okay, so you're measuring things to the nearest 0.1. What one had the greatest change in mass? I think we can have a look there. We can see that that is D. and then one here where they've just given you the equation. Again, I'll just work it through quickly, but hopefully these are some of those skills that at least you can follow through. Always show your working out, please, because you will get marks just for sticking it in the right place. So this is a 3 marker. So you get 1 mark for saying percentage change would equal the change in mass. So minus 1.1. Mass at the start, 6.0 times by 100. You get a mark for that. You then get another mark for sticking that into a calculator. This might actually be one where I can't quite do it in my head. Hang on. Oh, no, that's annoying. It'll be like 0. What, something. Hang on. Right, 1.1 divided by 6. So that would be minus 18.3. Yeah, it's on higher as well. I mean, like lots of these stuff like crossover, right? Like higher and foundation are not like things which don't exist, right? Like separately for each other. They overlap. It's just sometimes the questions are a little bit harder about them. That's all. And now I've already rounded it to 1 decimal place, but rounding it would then get you that. So if you just wrote out from the calculator, that'd get you a 2nd mark. Right. Next page. Screen closer. I mean, it's good. You're in year 10, you'll be fine. Oh, not a 5 U. Well, that was a hard pivot, wasn't it? Nah, if you've got a U, then you're screwed. No hope. That's a joke. Right. No, this one, again, it's an estimate. So can you see that you get here, right? Here, the potato is getting bigger or getting like heavier. Here, the potato is getting like lighter. So in between there, we have to switch over. This is what I was talking about, where we're in that bit is lies this. So here is where the potato is getting heavier. That's like the ones at the top. And here the potato is getting lighter. That's like the ones at the bottom. So there must be a point in between where it kind of is not doing anything. And as you can see, that's roughly plus 4.8. That's minus 4.4. So it kind of, you're probably thinking, right, it's going to be right in the middle of there somewhere. So let's guess 0.2. Now, whenever it says estimate, that means you have in the ARC scheme a range of what you can do as your answer. It's not you've got this, you're not 100% sure of the number, therefore no marks for you. It is just a case of having a look. Right. Cell division. Now, another part of cells we haven't talked about, but I thought I'd go over it in terms of when we do a question. Way to memorize them. I mean, there's only 4. That's lucky. Should we do that now? Let's take an aside. Let's do that. Food tests. Okay. As always, like I want to respond to you lot. So if you have anything, mark it in. Right. Let's zoom back out. Right. Food tests. Is it food? Yes. Right. So there are 4 food tests. We'll do it. B2, I want to probably do bioenergetics potentially first, unless anyone cracks in, because I did a poll at the start. Right, so there are 4 food tests that you need to worry about. So that is testing for starch, testing for sugar, okay, testing for protein, and testing for fats. Right. Now, starch is the classic. Okay, starch, we use iodine. Okay, now iodine is orange. Okay. Now, when I add it to starch, it goes kind of a dark blue, black. Okay. That is like orange to blue, black. Okay. I don't know, think of a way of remembering that. Orange, blue, black. So if I don't have starch, it will stay orange. If I do have starch, it will go blue, black. Right. Protein, and these two are the ones which are most easily confused. And I'm going to guess that that's probably the ones which you most often get mixed up. Protein is burette solution, and sugar is Benedict. Okay. Now, is there a way of remembering that? I'll be honest, I'm not sure I've come up with a really good one. No, but apparently she's living rent-free in your head, so I'm sure you can imagine. Anyway, if you're in year 10, like, go and enjoy your evening. Why would you want to tune into my boring voice? So with burette, okay, maybe you could say it's the same number, like it's similar number of letters. It's closer to that. I don't know. Benedict. I'm just going to be coming up with blanks. I generally just think that that's just two things you just need to kind of like try and commit. I would say those two are the ones which are easily kind of got a little bit confusing. Now they're both blue, which also doesn't help. So this one, you just put it in, you put the blue in, and it goes purple if you've got protein. Now the Benedicts, you put it in, so it's blue, and then you warm it up. Now what happens is it will start to kind of go like orangey if you've got some sugar. If you've got a little bit of sugar, it might go a bit yellow. Okay, just showing off all the colours I've got next to me. Right, and the end goal is like kind of a real red colour. Okay, so you kind of progressively get redder. Like red is the kind of end goal. Either way, look, Benedict has got Ben in it. Imagine there's someone called Ben and he's really sweet. Sugar sweet. We'll roll with that. Sweet Ben. And finally, you've got fats. Fats, you add some ethanol and you give it a shaky shake. And you will get an emulsion. Yep, brick red. Don't get a... Just say red, it's fine. Like, brick red is like... It's one of those things they like to talk about for like the flame test later on. Brick red is technically like correct. Like when they say brick red is technically better. Red should do fine at this point. Okay, those are the food tests. Cool. Yeah, both, because like they're all the same. If there's something which is higher only, I'll let you know. Okay. Anyone got anything else they want to go through before I do some more exam questions? You can have a 24-hour time, mate. Why not? Go and enjoy your evening. No, you're not cooked. If you were cooked, you wouldn't be alive. Got to be raw. Okay. So that's good because I was going to talk about bioenergetics. Let's do bioenergetics. Bioenergetics is one of those things where it's actually like quite straightforward. All right. There's basically like two equations, right? And that is, do you know photosynthesis and do you know respiration? And they're basically the same thing anyway. They're both in disguise. Right, where are we? Here's all the stuff that I printed out. Look at me. Look how prepared I was. Not apparently prepared enough. Apparently not prepared enough to actually do a respiration question. Lunatic. Yep. Do as many questions as you can. Right, so photosynthesis. Right. Okay. Fundamentally, it is how plants make everything. Okay. It is everything a plant needs to live and survive. And what it does, I have 3 YouTube videos. Oh, no, don't Cognito. Just do Cognito. There's loads of free Cognito. Right. So, what we do is the plant takes carbon dioxide from the air, and it takes water from the roots, okay, and converts it into glucose and oxygen. Okay. Now, what does it need to do that? It needs sunlight. Okay, it needs energy, and because it needs energy, that means that this is an endothermic process. That means it requires energy. Okay, takes in energy from the surroundings. To harness that energy, we need chlorophyll. And this is why plants are green. Okay. Now, with that chlorophyll, okay, we absorb that sunlight, and this makes that happen. Now, I haven't really seen them on Foundation asked for the formula, the balanced formula version. Okay. Okay. Yeah, of course you can. So with that there, the only thing to remember with it is it's an absolute devil to balance. So you can go 6, 6, 6 if you're ever needing to do that. I think that'd be really mean. I don't think they'll ever do that. But that's it. Now, what happens with photosynthesis? Well, there is a required practical to do with photosynthesis. So that's looking at how can I affect these different things. So, for instance, you often, you might have done this with, it's the only time I ever see these used in school, right? Like the lamps which look like Pixar, right? Like those ones, right? You know the ones I mean? Look at that. Art. Okay. And what you do is you say put some pondweed in our test tube. Now we cover it up with not water but sodium bicarb solution, carbonate, and that's our like kind of source. It's like almost like dissolved CO2. Okay, we Look at that lovely ruler, okay? And we put the light at different distances and we see and we count the bubbles, okay? That hopefully might kind of jog a memory. Now, remember, whenever you're doing a required practical like that, do numbered lists when you're doing a method, okay? Say what you're varying. So what your variables, what you changing each time, okay? Don't forget to mention repeats, okay? Doing it multiple times and calculate mean. And if you can draw a diagram, draw a diagram, right? If I was an examiner and I'd seen this, like, and it's labelled, I know what they know what's going on, and they vary. So you can do variations of this experiment. So you could do it where I change the light intensity by putting the plant at different distances. Okay, I could do it by looking at the color of light. I could change what the bulb is. I could do it by changing the concentration of the sodium bicarbonate. So they're looking at how carbon dioxide affects it. I could also do it with different temperatures. So I can have the whole thing at different temperatures. Okay. All sorts of different things that you can do with it. Yeah. Yeah. If it says give a method, you're still going to need to give a method. But like, if there's nothing wrong with drawing pictures. You push it to 60 by, like, you know, not calling people unk, maybe. Who knows? And I'm old. That makes sense. Just do past papers. That's all you got. Right. The time to answer that question was like a month ago. The second best time to answer that. I know. I'm being silly. I'm having a joke back. Roll with it. Yes, all right. That would be the way of doing it. Okay. Right. So I can change my variation. I'm not going to do a whole past paper now because I've literally got videos where I've done like whole biology past papers. So you can do that with different ones like that for photosynthesis. Now, the other thing, right, is respiration. Now, if you can remember one of these, so photosynthesis, carbon dioxide plus water goes glucose and oxygen. Here's the cool thing about respiration. It's exactly the same thing, but flipped. Okay. I was probably going to go through to 9. But we'll see. Depends on when I get bored, if I'm honest with you. Right. So, photosynthesis, like flipped, is respiration. Specifically, aerobic respiration. Okay, so if I take that, I add glucose plus oxygen this time, so that's the other side of it. Okay, and then I get carbon dioxide and water, right? Okay, now whereas with photosynthesis, like it required energy, with this, with respiration, it is exothermic, it gives that energy, which is the point of it, because you want it to provide energy for your cells. What plants need starch and cellulose after making it from glucose? They need, so glucose is like the first building block. So you're then putting that together, right? It's kind of like if you are like making a Lego set, like this is your like basic block to then add things to and then make whatever interesting thing you need. So you need glucose. So this is now taken aside. What are the uses of glucose? Well, first of all, you can use it for respiration. and using it for respiration is how we're going to get our energy back. Remember, all plants need energy. Every living thing needs energy. Every living cell needs to do respiration. If it doesn't do respiration, it is dead. So first use glucose is doing it in here. And because of that, if I can't use it all the time, okay, we can store it. Now, the problem with sugar is it's soluble. So if I'm not careful, I'll constantly be losing some. So what we do is we make it insoluble, and they do that by turning it into starch. Okay. The other thing we can do is we can use it to start building things, right? Now, specifically, we can make it into cellulose, which is actually, if you ever do A-level degree, is basically chemically identical to starch, just depending on the type of glucose, alpha and beta glucose. Don't worry too much about that. But the cellulose, we can use in that for cell walls. Okay, we can combine it with nitrates, and now you're starting to make like amino acids, and then you're using that to make protein. Like, the glucose is your starting point. You use it for energy, you store it, or you make stuff. Those are the three things you do with it. Okay, right. Can I explain the required practical? You've missed that. Have a look back if you need to. Right. Back to respiration. So we're taking our glucose, we combine it with oxygen. Okay, we get our energy out. Now that is all well and good. Okay, we're going to get, that's the whole point of it, is we get energy. We'll just put it in brackets because it's not stuff. Okay, now this is aerobic respiration. Why? Because it requires oxygen. Okay, that's what makes it aerobic. Now, you need to do that, as I said, all the time. Sometimes that's not enough, right? So let's say I'm doing a 100-meter race. Okay, and in that 100-meter race, by the marvels of science, a saber-toothed tiger pops out from behind the start and decides I'm going to chase him down. Now I'm going to be really incentivized to actually peg it. So what happens then? I'm always activating my inside boost button, and you do this all the time. Okay, you do this all the time where if you're working beyond your capacity, you start to go into your added boost button, if that makes sense. And that's because, right, this is happening all the time. I need to get oxygen in to do this, right? So as I start to work harder, because I want more oxygen, I will breathe faster. Okay, my heart will pump faster and with greater volume. And I'm sending that blood around, okay? So what's the point of this? That means there's more blood and more oxygen in the blood, right? Breathing faster means I get more oxygen in there, okay? Heart pump faster means that blood's getting around, right? Eventually, that still might not be enough, right? You're still doing that at full whack, right? What you can do is and this is, where you're leaping ahead with lactic acid. You can do it where you don't use the oxygen. You just kind of skip it. And this, instead of going to carbon dioxide and water, this goes to lactic acid, okay? And it gives you some energy, like 15 times less energy. So, because often people say, like, if I can do it without oxygen, why bother? Why don't I just do this all the time? I could breathe I don't even need to breathe underwater or anything. I just chill, right? Now the problem with that is this does some energy, not a lot, and also that's bad, right? It starts to give you like cramps. Okay, it makes change your blood pH. Which makes other stuff happen, means it's not good. So this is why I like to refer to this as our extra boost button. Without oxygen, we call that anaerobic. You cannot sustain that for long, okay? You know this, right? If you're running, you're like jogging, you're doing this, right? chill. Maybe you're doing a little bit of this, so over time it starts to really build up by the time you get to the end of the run. But if you do a sprint, you go straight onto all of this, right? So by the time you finish, like if you're doing like 400 meters or something, you get to the end and you're like breathing heavy and your muscles are like screaming. Okay, now why do you keep breathing heavy and all of this stuff? Why does that keep it going for afterwards? That's because that lactic acid we don't want, so it wants to go back to that. To do that, we actually have to get the oxygen back in. And we call this an oxygen debt. So this is kind of like, you can think about this as the oxygen that is kind of owed. All right, so if I get that oxygen back in, I will convert that back out to carbon dioxide and water. So that's why you keep a high heart rate or higher heart rate. It will slowly start to come down, and a high breathing rate. Okay. Is there anything else that I'm missing? Oh yeah, plants don't do that one. So plant version of this, also yeast does this. Yeast. What it does, it will take the glucose and it won't turn it to lactic acid. It will turn it to ethanol and carbon dioxide. Lovely stuff. Now, we use this. We use this because by making ethanol, that's like brewing beer and wine, right? So it does both of this, right? So if I'm more after the ethanol, then that would be brewing alcohol. If I'm more after the carbon dioxide, that's for like bread. So you get the bubbles and it rises and it's like nice. It's not like a dense puck. Cool. That is the bioenergetic stuff. Anyone got any other questions? I know you said heart, so I'm tempted to do organisation next anyway. There's a heart lurking there. I haven't really talked about specialist cells yet. I might go back to specialist cells actually. Right, I'm going to take a drink to pause. Anyone need anything in particular? It's quite a noisy bar actually. Who is it in the Wizard of Oz who needs a heart? Is that Tin Man? Or does he need a brain? The lion, does he need a heart? Or does he need courage? Why can't I remember the... Is the Tin Man? Of course it is. I am a Tin Man. Right. The cast of The Wizard of Oz will be on your test tomorrow. That is true. But Wicked is higher only. So if you don't know who Elphaba is, that's higher only. If one of you now goes and revises Wizard of Oz, my mission has succeeded. Right, let's look at the heart. Can I say, Wicked being higher tier only has been my favourite joke so far. Right. Right, we're in play. We're in place. I'm not going to try and do that. Also, you fail your physics if you do Wicked because you're defying gravity. Right, let's go. Heart. Right. First of all, okay. Shall we go? I tell you what, we're gonna go rogue here. Oh, nothing revealing behind me, I don't think. Oh my goodness. Look at that. Right. Hello. So, oh my goodness. Right. With the exam paper for the heart, before I hate my face, right, here we go. For the exam paper for the heart, literally pick it up and put it on your chest. Right? If you pick it up and put it on your chest, you get these sides the right way round. Okay. So the left-hand side is, if I pick it up and put it on my chest, is there, which when I look at it, wow. Oh my goodness, what have I done? Put this slide cover on. Whoa. Right. So when I pick it up and put it on my chest, that what looks like on the right of here, okay, is actually the left. And what looks like the left is actually the right. Okay. And it's because people drew this by like opening up people and then looking at it, right? So it's why it's mirror flipped. If you lift it up and put it on your chest, like, yeah, you might look like an idiot, but you'll be a right idiot, and that's good. So those are them. That's the way to get it around. Other way to get it around, okay, is that it's thick. CK, not double C. Okay, it is thick. Why? It has got large amount of muscle. Okay, because it has got a pump. The left-hand side goes to the whole body, right? Whereas the right-hand side is only going to the lungs. Okay, so it doesn't need that thick muscle to only needs to pump it a little way. So it doesn't need to it doesn't need to go far. Right, what are the chambers called? So you have top chambers and then bottom chambers. Now the top chambers, you might have heard of these maybe. The Latin word for entrance hall is an atrium. And if you ever go into a fancy building, they might call it an atrium, right? The room you go into, right? Like a lobby. And that means entrance hall. So if you think about it, like the blood's coming in, right? So it's going to, first thing it's going to go is into those chambers. So I've got the left atrium and the right atrium. Right? Now, from the atria, we go to the ventricles. Hello, Tiggy, how are you? Do you want to meow for the live stream? Oh, you do? You're good. Oh, okay, that's a hiss. Everyone's going to think now you hate me. That's really mean. Do you hate me? You do? Okay, that's fine. Right. So, the ventricles, okay, so the blood comes into the atrium, pumps into the ventricles, and then from the ventricles we go out. Okay. So, the left-hand side is all about getting oxygen into the lungs. So here we're going to get deoxygenated blood. Okay, and that has come from the whole body. And there we're going to then pump it out to the lungs so it can collect its oxygen. Here we're then it's gonna come around, it's going to lungs, lovely. And it's going to come back in here. Okay, so I've now got oxygen in it there, and then from here we're going to go out to the whole body. Now these blood vessels have names. So we've got the atria and the ventricles, we've got the valves. Okay, this is called the vena cava, and this is kind of like the biggest vein in the body, as it collects all the blood from the body coming in there. This here, because it's going to the lungs, this is called I'll write it up here. It's called the pulmonary, because pulmonary means lungs. And because it's leaving the heart, it's a thicker blood vessel, needs to deal with the pressure. That is an artery. Okay. Then it comes back in here, so it's come back to the heart, so it's pulmonary again, because it's come from the lungs, but it's a vein because it's returning to the heart. And then here, emanates the body, that's called the aorta. Okay. Right, so the questions, I see you've already given it a go. Beautiful stuff. Okay, which part of the heart receives oxygenated blood from the lungs? Okay, so we've come in here from on the left-hand side. We've got that blood in from the lungs. That makes it A, the left atrium. Which part of the heart pumps deoxygenated blood back to the lungs? Here, I've got the deoxygenated blood back to the lungs. So that is actually C. Okay, right. Turn it up. The cha-cha slide still a thing? Like, would people play that at like a disco? Not that people have discos. I feel like it only comes out of weddings now. That's the heart. Any other questions before? If not, I'll just kind of crack on with some stuff. We've got infection response. I haven't done cell division. Those are two things which I knowingly haven't done yet. If anyone wants to request anything in particular, but that's the heart. Does that answer some of your questions on the heart? Cell division. Let's dance to cell division. All right. Okay. So there are two types of cell division. Now this is where we do start to get a higher and foundation split. Because my classes are technically foundationalists here, I'm not going to go bonkers on the higher, but I might just talk about it briefly. Okay. So there are two broad types of cell division. There's mitosis and there is meiosis. Okay. Now, mitosis is the type of cell division that you do for growth and repair. You are doing it all the time, right? I've got cuts all over my fingers. Okay, my body is like, I need to fix that. Oh, shiny. Like, I want to fix that. So it will be doing that. We're doing it all the time to kind of refresh, regenerate, and repair. Meiosis is to make gametes, which are the sex cells. Okay. Now, I've always thought about this. Like, imagine someone came in and just like, because cell division is quite logical, right? If someone just chucked like a ton of paper at you, like a book, right? But they've cut the spine out, so they just chuck you a ton of paper. Nice. That's how I roll. So I just chuck the entire like pack of paper at you, and I say, I want you to copy that whole thing. Right? I want you to make an identical pile. Well, the first thing that you do, okay, is you take all the stuff, right? You take all the stuff. So there's all my papers, and I would copy it. Right? So the first thing I do is I copy it. The thing is because I want to make sure I do it properly, what I'm going to do is I want to make sure that I sort it out into the two piles really nice and neatly. So what I'm going to do is I'm going to line it all up. Right, in my two piles, ready to go. And I'm going to check each one. And when I'm happy, they're going to kind of go, and I'm going to split. Okay, so copy, line up, split. It's not the technical term, right? It's not. Okay, they've technically got phases. So you've got like, we've got these phases, anaphase, all these things. Don't care about that, right? I want you to get the idea of what's going on. We copy it. We line it up, we split. Okay, we copy, we line up, we split. Right. So if I look at this question, right? Can you see here? This is my picture. It's quite hard to see on here. I've got these cells, these nuclei, just big and chilling. Okay. And can you see here? It's hard to see. It's much clearer on the actual one. Right, you've kind of got these like chromosomes like this, and they're starting to be pulled apart. And here you can see they're really pulled apart. Okay, so these two are undergoing that cell division. These other ones are chilling. So if I have a look, right, I've got 8 cells in total. Right, now 2 of those 8 are dividing, which means it's a quarter. Okay, that's how it rolls. Okay, now again, let's track how many chromosomes you would have throughout. So I like to talk about this. Normal human cell has 46 chromosomes, 23 pairs. When I replicate, I'm now up to 92. Okay, I'll line up, I'm still at 92. I split and I've got 2 identical cells, each with 46. Okay. Now, obviously, I could draw this much better, right? And you see them in the nucleus, and you see them in the spindles, and they pull it, right? But I'm just kind of focused on the night before, right? I'm not here to kind of like teach it to you like fancy. Now, meiosis, okay, because this is for sex cells famously have half the DNA, right? Because if they have the whole amount, not going to work. Okay, want to have half from mum, half from dad. Lovely stuff. So what they do is we start off with our cell, and actually, weirdly, they copy as well, right? It does get more complicated. At this point, they actually can start to like kind of switch up, right? They actually kind of scramble. This makes, because you want variety, right? This is the point of sex cells, is you're wanting variety so that you're increasing variation. And I know this is more B2, but by having variation, you're going to have the chance of something being more adapted. Okay, so this does come in a little bit more into that B2. So don't, it kind of straddles a little bit. So we're here, and then we actually divide twice. So we split twice. So we started with a thing. Okay, so we started with our 46, we're up to 92. Now we went with 46 again, and now we ended up with 23. So here we started with one cell and ended up with 2. Here we started with one cell, we ended up with 4, but they got half the amount. Okay, right, let's put it into action. So we've got our division, right? Okay, it says the cells which are not dividing each contain 10 chromosomes. One of these cells divides by mitosis to produce 2 new cells. How many chromosomes will each new cell contain after mitosis? So the whole idea of mitosis is you end up with identical cells, right? So I start with 46, I end with 46. It's like you just popped out another one. So if I start with 10, I end with 10. Okay? Cells divide in a series of stages called the cell cycle. Choose its answer from the box. What question may I ask? It's this one. Okay, so before mitosis occurs, the cell, okay, what does the cell do? The cell replicates, doesn't it? Well, it actually grows first, right? You want it to get bigger, actually. You want to get bigger, so you've got space for 2. The genetic material in the cell doubles when the DNA replicates. After the chromosomes have been pulled to each end of the cell, the cytoplasm divides, so it kind of splits off. Okay, and you can see here I've got different stages, okay, as it's going. So which stage of the cell cycle takes most time? Again, it's not after anything complicated there, it's just to see, because that's the biggest part of the pie. And what percentage of time is it in A? That would be 10%. Now, I've cut off the question, but I want to talk about stem cells. Okay, now, let's get a bit of paper. Right, stem cells. The key to the thing about stem cells is actually to think about non-stem cells. Now, that's not a plant pun, right? Because stem cells are actually nothing to do with like stems of plants, apart from you can have meristem, which are the stem cells in stems. Blah, right? This is a specialized cell, right? That is not a stem cell, it's a specialized cell. Why is it specialized? Right? Because it's got like features which make it good at this function. Right? It'd be like, I don't know, me growing a massive hand, so I'd be incredible in goal. I don't know what you're thinking, sir, you're already incredible in goal. But like if I had 2 massive hands, I'd be phenomenal. Right? Or being able to just have like 2 long legs, so I could just like, I don't know, do like long jump or something. Cells have that flexibility where that they have different structures, which makes them good at the job that they are meant to do. So here, I've got a cell What type is it? This is a root hair cell. Okay, and the question is, what makes it good at its job? All right, what makes it good at its job? Well, it has got, okay, this long hair, and that has got a large surface area. Right, what we were talking earlier with transport, by having a large surface area, you've got the ability for things to enter and leave the cell. Okay, it has mitochondria in there. That's what all the dots are. Or it might be my printer. Who knows? And the mitochondria are there to provide energy. Everyone has them, but they'll have more, so that energy can be used for active transport. Okay, so you can see I've got a couple of adaptations there. The classic one that everyone talks about is a sperm cell, like because it's got a little tail, so it can swim and find the egg. It's got mitochondria to give that energy to do that. It has got like what's called an acrosome at the front, which means it can burrow in and kind of fertilize the egg. Its nucleus has got half the amount of genetic information. Here's another one. I've got a nerve cell. So the type of cell is a neuron or nerve cell. Now, what makes this really good? You've got lots of places for connections. Okay, so you've got dendrites and stuff to make connections. You have got this really long. Okay, it's called an axon, and that means that basically I can send a signal from one side to the other really long. It's also got these kind of these bits here, which provide kind of insulation to stop the kind of electrical signal leaking out. Right, so it's well adopted to give a nerve signal. All right, so you've got all these places to connect with other nerve cells on both sides. It's really long. You've got this this kind of insulation to stop the kind of electrical signal leaking out. And that's an adaptation. Right. So that's specialized cells. So what stem cells are, and stem cells come in two types. You have embryonic and you have adult stem cells. Okay. Now, adult stem cells might be like we'll have skin stem cells. You might have the classic place for stem cells is your bone marrow. And the point of these is they can do our mitosis, and they can specialize, and they can make a limited range of cells. So, for instance, a skin stem cell is not going to make my heart. Okay, my bone marrow stem cells are not going to regenerate my nerves. Okay, they can make a limited range of cells. Now, embryonic can literally make any cell. Why? Because where's an embryo? That is our developing baby, basically, right? That's when sperm and egg meet, they've divided a few times. So, of course, it has to make every type of cell because it's going to end up with a baby, which is every type of cell, right? So, you need those types of cell to make blood cells, bone cells, muscle cells, all of those types of things. Okay, so these stem cells are undifferentiated or unspecialized cells. Okay, and then they become specialized through this process of differentiation. Embryonic can make pretty much any of them. Adult stem cells, they can make a limited range. What do they like to talk about? They like to talk about and ask about like treatments, like if the advantages and disadvantages of different types of treatment could, so like they like to sometimes talk about the moral aspects of it. So embryonic stem cells, in theory, come from embryos which could be implanted into a woman and make a new baby. So they like to talk about those kind of moral aspects. But the key thing really is to think about stem cells as undifferentiated cells. They can make embryonic ones can make any type of cell. Adult stem cells make a limited range of cells. Any random questions at this point, or any topics that you would like me to cover? So I'll go for another 15 minutes. Unless you're all bored. Should we do some infection response? I think that's the one we've got left. 1-1. Right. What's an absolute mess of paper? It's a beautiful cell. Well, I'm still that. That's a good one. Right. That's a bit of tidying. Right, let's do some infection response, shall we? Let's crack in with some questions. Why not? Everyone loves a question. So, first thing about infection response is remember what a pathogen is. Why am I using the most boring colour? Let's zhuzh it up. Laura, you'll have an interesting colour choice. Right, so pathogens. These are things that make you ill. Okay, green, cool. Right, we're subbing in. I'll use all the different shades of green that I've got. Got that one. Oh, look at that. That's a bit mucusy. Oh, it's perfect for pathogens. Love it. Right, that's our mucus colour. When I mention mucus, we're subbing in mucus. So there are 4 types of pathogen. There are bacteria, virus, okay, fungi. I know I am, but they also infect you. And protist. Okay, protist is a single-celled organism. Right, you need to know some types. So, salmonella, food poisoning, is caused by bacteria. Because when the sun shines, we shine together. Bacteria causes salmonella. I don't know, do the rest of it. Any tips from Oro? Pray. No, don't do anything stupid in the nicest possible way. Like, do your real solids. So, like 10 minutes before the end, just think to yourself, sanity check, right? Sanity check. Go back. Have you ticked 2 boxes, right? Have you filled in like any missing like words? Is there like any questions which don't have an answer line, and because you were rushing, you didn't read them? Like, there's actually a secret question that you've missed, right? Have you like turn over and check that it gets at the end of the paper? It says end of questions, right? So do check that you have got to end of questions and not you're missing a page. If it's like a graph and it asks for a line of best fit, do the line of best fit. If it says your answer must include bullet point, bullet point, bullet point, cross them off as you do them. Okay. Cheers. Well, I just don't upload enough because I work as a teacher. So like, I'll be honest, I love science, but I also don't want to get home at the end of the day and go, you know what? I want to do an exam paper. Love that. Right. So, salmonella, gonorrhea. It is on your exam specification that you need to know that gonorrhea causes yellow-green discharge. That is on your GCSE. Virus, things like HIV, tobacco mosaic virus is the one that they love, okay, because it infects a plant. Fungi, athlete's foot. The other classic that they love is rose black spot, which, funny enough, infects plants and gives their leaves black spots. And the classic protist disease is malaria. Okay. So, how do we? Malaria is so you might. Right, brain's going. So, you might need to think about how these are spread. Okay, so are they spread through the air? Are they spread through water? Are they spread through sexual contact? Are they spread through mosquitoes, or what we call a vector? So because these go via malaria, malaria via malaria. Because they go via mosquitoes, the mosquito is what we call a vector, not the baddie in Despicable Me. Or isn't there someone who dresses up as Vector and starts doing like random reviews for some reason? Rogue. It's all right. I've got the bowl cut at the moment. I need to have a haircut. It's looking shocking. Right. So, how do you defend against them? How do you defend? Well, let's do a defensive barrier. Right. The first thing is that you have skin. Right. I'm going to probably go for another 10 minutes. I don't want my wife to think that I hate her. And I've actually got to prep my lessons and stuff. So I've got skin. That is a physical barrier. Stops anything getting in. Lots of people start going and talk about scabs. That's what happens when there's a break in the skin, and you kind of whoop, and you stop wobbling. Right, and you stop things getting in. But skin is our good barrier. Out comes the mucus. Right, this is going to stop any and trap any. You can tell how much I've used this. Look how thin that line is. This is going to trap any bacteria in in any membranes that are coming in. But let's say that they have breached the barricade. They're coming in. Right? There's other things like hair. Oh, stop hitting the camera. Things like hair. Things like tears. It's all of it. It's both. Because everything on foundation is naturally on higher. So we're in. Okay, we're now in. I mean, we don't want to be in because these are the baddie pathogens. So now in come the white blood cells. Right, and these work in 3 ways. The first way is technically called phagocytosis. Let's put that in for those higher beans. Phagocytosis is a fancy word for engulf and digest. So what happens is you've got a little bacteria or something. Right, here's my little bacteria, right? And my white blood cell is like, here it is. Here's my cell membrane, and the white blood cell kind of goes whoop, digests it. Don't say eat. I will not accept that. Okay, so you're physically kind of consuming the white blood cells. Not consuming the white blood cells. You're consuming the pathogen. Okay. Right. Next thing. Okay. So is bacteria. Half the reason they make you feel ill is they make toxins. Okay. And white blood cells make antitoxins. And that helps you feel a little bit better. It's blocking out the haters. God, I really have lost it, haven't I? And the last one and the most interesting one is making antibodies. Okay. Now, antibodies are amazing. Okay. They basically are specific to that. So they are specific. And they will bind onto the pathogens. They'll get them all clumpy and stuck together, and then this engulf and digest thing can kind of mop them up. Now, the thing with antibodies is they take time. Right? Because basically, you've got to kind of work it out. Right? The analogy that I kind of think of is imagine your job is to crack a safe, right? You're like a safe cracker. The first time round, right, it's going to take you time. Okay, takes you time to work that through. Okay, you're going to have to get your thing out. Tiny Tim doesn't die. Pretty sure, right? Anyway, that's done. Christmas, whatever, done. Lady Macbeth washes her hands, done. So you've got your stethoscope and you're like listening to the safe and you're like picking it all. That takes like time, right? And then you're in. Now, if you get given that exact safe again, you just use the combination again. It's really quick. Okay, so it takes time to work it out, right? Now, if it's a bad disease, you might have died in this time, right? You might have died in this time. Okay, so the time it takes to get from to you to work it out, okay, it doesn't. You famously at the end, doesn't he go like "Merry Christmas, everyone"? Anyway, so it takes time to do that. But if I now have it, I am then going to be really fast next time. And because I'm really fast next time, you are basically what we call immune because you remember how to do it. Okay? Oh, okay. You really do need to pray before this exam. I'm not even religious. Okay. Do you know that Scrooge actually was actually Donald Duck in disguise, or Michael Caine? Okay, bye. So yeah, takes time to work it out, but it means that next time you're really fast on it, and that's because of something called memory cells. So if it's a really bad disease, that's where vaccines come in. And a vaccine is where you're injecting an inactivated virus, inactivated pathogen, gives you an opportunity to kind of train on that. Okay, gives you an opportunity to train on that. And then when you get it again, you go like that. How do spores affect the roots of the plants? You're thinking of nodules. So they are bacterial colonies which help fix nitrogen. So that means you've got high nitrate in the soil, and therefore it can do it. Right. So, or I suppose if you're thinking about fungi like that way, you're going to compete for resources, and therefore it's not going to work from that perspective. So you can also if you're doing it with spores in terms of fungi, in terms of fungal infection of plants, it could be like that. So with a vaccine, you'll get an inactivated pathogen. Your body will then make the antibody. Okay, so they make the antibody, and then if you get the actual disease, the actual pathogen, you've cut out that time step. So it's now really fast. Okay, so when you get the actual pathogen, you're then there. So often questions like this will tend to have like a graph, right? So let's say this is antibodies, okay, and this is time, right? Let's say you got the vaccine, right? You have this lag period as you work it out. Okay, so that's our kind of lag period. And then here we've worked it out and we kind of have done that. Now, let's say here I get the infection, right? So I'll get COVID straight away. So you get a much reduced kind of lag time, and you can do a really fast, rapid, and big response. Two other things to do with this. First of all, you've got antibiotics. Now, antibiotics only work against one thing. They work against the bacteria. Okay. And the other thing is viruses are really annoying to fight. Right? Why are they annoying? Don't write in the exam. They are annoying. Okay. The way that they reproduce is they will take your cell and the virus will go and infect your own cell. Right? They'll take it over and start making copies of themselves. Okay, and then they'll burst out. Right? Releasing lots of virus. Now, why is that annoying? Because when they're inside your cell, right, from the outside, you're not going to know that that's infected. So it's really hard to get drugs which will treat the viruses and kill the viruses, but not kill your own cells. Because at the end of the day, if you kill your own cells, yeah, you'll cure the disease, but you will also be dead. So that's why antiviral drugs are much harder to work. I think we have weirdly covered pretty much everything. I'll do a any last questions, basically, before I suggest that you chill, because you don't want to be like tired. Any final questions? Obviously, there's lots of stuff that I wish I could have probably gone on over in a bit more detail, but it's not going to be like I can't teach the entire course in an hour and a half. I say, if there is nothing, I'll bid everyone adieu. I believe that's how the French say it. Obviously, my lovely lot, good, Delaray. That makes a change for you because you are really harsh on yourself. So I'm glad that you feel pretty solid. If you have any questions, obviously, if I actually teach you, don't hesitate to email me. I'll reply and stuff. So like, yeah. Yes, making new drug is on tomorrow. So drug trials, they start on cells in the lab. Then they might do an animal trial. Then you'll start doing human trials. Okay, human trials, you have different phases. So phase 1 is on a small number of healthy people, just checking the drug is not going to kill them. Then you do phase 2, that is on the actual ill people. And that one you're working to see the dosage. Then phase 3, this is a large trial. Okay. You've been attacked by a banana, young man. Okay. Then phase 3 is a wider trial with lots more people. And that is checking to see if it actually works. In that case, you test it against either a placebo, which is a drug which should not do anything, but will do because the placebo effect. Or you actually test it against the next best drug. So if you're looking for a painkiller, you wouldn't test it against placebo. You test it against like paracetamol or ibuprofen. And that is also called a double-blind trial because the doctor doesn't know if they are giving them the actual drug or if they are giving them the placebo or previous drug. Cool. Right. Have a lovely evening. Bye.